Hello everyone, I'm Rhianna Lilly-Smith from Technology Networks, and today I'm delighted to introduce Dr. Jeremy Barr, an Associate Professor in the School of Biological Sciences at Monash University, to discuss their latest study published in PLOS, which investigates the interactions between mammalian cells and bacteriophages, and how these viruses may be offering a benefit to their mammalian hosts that goes beyond killing bacteria. What led you to undertake this study, and how did previous research influence your decision?
So our research out in PLOS biology looks at the interaction between bacteriophage, which are viruses that only infect and kill bacteria, and mammalian cells. And so our inspiration for this research really stemmed from some of our prior work where we showed that bacteriophages, these bacterial viruses, do in fact interact with diverse mammalian cells, including cells from humans and also other organisms. And we found that these mammalian cells will actively uptake, process, and traffic these phage particles and move them around throughout the cell structure. And this is of broad interest for a number of reasons. Firstly, phages are prevalent within your body, within your own gut, and actually all distributed throughout your own body. But on a more broader context or perspectives, we're actually using phages to treat bacterial infections in a process known as phage therapy. And so the dosing of large numbers of phages into the body raises the question, what are they doing and how are our own cells responding to these viruses?
Can you explain the main findings of your study regarding the interaction between T4 phages and mammalian cells, and how it impacts cellular growth and survival?
And so what we found from our study was that phages did in fact interact with these mammalian cells. And the first thing that we demonstrated was that these phages were not causing an inflammatory immune response. And this is really important. It suggests that these phages were safe to administer to humans or animals, and that they were not causing an inflammatory immune response. The second major thing we found in our study was that these phages were causing broad cellular changes, a lot of these protein phosphorylation cascades that were leading to an increase in cell growth, proliferation, and metabolism. And this suggests that these cells are internalizing and uptaking these bacteriophage particles, and that they're using these phages as a resource to promote cellular growth and proliferation. And so we show one of the first pieces of evidence that there may be beneficial impacts of applying and dosing phages to mammalian cells.
Your study suggests that T4 phages trigger signaling pathway events that promote cellular growth and survival in mammalian cells. What do you believe is the underlying mechanism for this phenomenon? And are there any specific reasons or advantages for cells to use phage particles as resources?
Now, before I go any further, it is important to note a few caveats for this study. This work is all in vitro. We haven't applied this to in vivo or animal models as of yet, and we've only looked at a single phage type. So there's much more research that needs to be done here. But we've also provided some insight into the underlying mechanisms here. We believe this is a really pervasive and diverse mechanism and suggests that every and all cell types are capable of interacting and uptaking these phage particles. And that the mechanisms here are thought that these mammalian cells are interacting, engulfing, and trafficking these phage, and that using them as a resource to enhance their own cellular growth and proliferation. This provides the cell with potential benefits. The cells are constantly exposed to diverse phage particles, and this may provide a broader advantage to these cell types or to these hosts.
In light of the increasing use of phage therapy to treat antibiotic resistant infections, could you discuss the practical implications of your research and how it might influence the development and application of phage therapy in the future?
Now, within the context of phage therapy, this is a really interesting concept because as a field, we are just beginning to treat a wide number of patients with different and diverse types of phages to treat some of these really horrible and horrific antibiotic resistant superbug infections. And it's important that as we begin to apply and dose patients with diverse phages, that we understand what is the cellular response and mammalian host response to those phage. Now, much more work needs to be done here, but our preliminary study suggests that phages are broadly or maybe specifically safe to use in this case, and that they do not trigger an inflammatory or immune response, and that there may be other broader symbioses that provide beneficial effects, at least to cell types under experimental conditions, that could be extrapolated to patients and organisms in the future. So a lot more work needs to be done here, but we're excited by these preliminary results. Thank you.
To make sure you never miss a story, subscribe to Breaking Science News, the daily newsletter by Technology Networks. Bye.